Hello, g'day. I hope you are all good today. Welcome. Oh, I've tried to put on I'm going to try and watch all of the comments today, so I should be able to hear you. Not hear you, see you. So I'm trying to get that organised on another screen on my phone here, so I don't miss any, so I can be interactive. So that'll help a lot. So when I skim through my stuff, people will see. I will see, and I can interact and help a little bit better. All right, I'm going to get started. So, all right, where's the slides? I've got to share screen. Share screen. Entire screen, I suppose. All right. So this is all the stuff we're going to go through today, all this stuff. The first thing was the poor Queen. Rest in peace, Queen. So I just want to show my respects to her. She's really cool. She's all I've known and my parents have known, and she's just really good. She's always smiles and always keeps a good friendly life. So I just wanted to show my respects to Her Majesty. All right, and now the microbes. So microbes are very, very beneficial. Unless you use your synthetic nutrients, you will need microbes to unlock your availables, because plants only take, like for instance, nitrogen in two forms. So it's your NO3 and your ammonium, your NH4, and your NO3, which is nitrate. So any other forms of nitrogen, it just won't accept. It'll just block it. No, it's not the right thing to uptake it. So it has to be transformed, usually through microbes, fungi, bacteria, stuff like that. And this is one process where you can do it through biochar. And this is a little cycle it goes in. So it starts up here and it absorbs onto it. So it's got all its minerals and things, and they absorb on, so they get attracted to the surface of it. And then that allows them to be transported around, and they're retained there. And then they keep building up as they get moved around through the soil. Earthworms, microarthropods, things like that will disturb them. And then it'll get put close to the roots where the rhizosphere is. And then that'll allow the exudates to react with them and the microbes and the fungi and exchange. And then it can be uptaken. So it's little how important the microbes are. This is another thing. Botrytis cinerea. That's the name of the fungi that infects and gives you the grey bud rot. And it was just a test done on tomatoes that says that from having an infection, it ended up accelerating the ripening and the cell wall disassembly. So it's sped up the ripening. So it's just an interesting thing, because we get that. I'll try not to get it, but if it does happen, it'll speed up so it must release ethylene or something, and it just speeds up to finish the plant. So it's just something interesting I thought I'd just throw in. And this is something really cool. So you know how greenhouse gases is caused a lot by methane? Well, it's carbon dioxide, but methane's 30 times more damaging than carbon dioxide to the greenhouse gases. And a new study they've done, they found out by adding a bit of seaweed to the cow's diet, it reduces the methane emissions by 82%. So that's virtually gone. That's so cool. That's just excellent. Using organics to try and stabilize the planet is the key. And the more people that are going to get into it soon, and it's just really good. So that's I just like that. Microbial inoculants and crop productivity. So this is how they can improve the yield average by 34% from this study. And they added some Pseudomonas, some Bacillus, and some Enterobacter. The alleviation of stresses. It helps with 54%. It reduced the stresses by 54% from putting these inoculants into the substrate, and improve the nutrient availability, because these microbes can transform it into its right elements and break it down to what the plant requires. So the yield gain potentially is huge, 34%. Pretty good. I hope I don't think anybody's. I'm going to stop sharing because my little thing. No, nobody's written anything. It's still working. Yep. Okay. I'll back to it. Slides. Shit, where was I? I was just sharing my big screen, wasn't I? Share screen. And I was on this section. Holy moly. Inoculants, that's right. Next one, what's this? Oh, this is how the Czechoslovakian Republic are decriminalising cannabis this week. So just Australia is, I just get shitted. That's why I put all this stuff in here, just to wake the government up. Like fair income. Are we going to be last on the planet again? It looks like it. So Czechoslovakia, congratulations. It's too much money involved in Australia. Oh yeah, but yeah, speaking of money, look at this. So Colombia has a rate, they do cannabis now, and they're exporting 18% of their total exports is to Australia. So we're buying cannabis because of our fucked up laws that Australia has with our government and all of our taxes. You know, it's like 70,000 to get inspections. I looked into the legalization because I'd love to do it. I'd love to grow it, but can't. No, because of the legislation. It's just too full on. And now everybody else in the world's capitalizing on Australia's shit government's rules. So yeah, 18% of Colombia's cannabis gets exported legally to Australia's market. This is a cool safe way to pipe, I suppose. You know, you're not going to combust the paper on the outside. You're only just going to combust your cannabis inside. So if you can't vape, at least it's a good alternative. I thought it was pretty snazzy. Nice filter on the end of it too. Just jam it full. You wouldn't put it like tightly packed because it wouldn't suck through, because that's pretty long. But I like the idea. I've never used it, or I don't think I've used a glass pipe. Oh, and there's a little empty one. That's like a little one hitter. Actually, I have used a one hitter. You put your little thing, yeah, that's a one hitter, because you put your little tiny thing in the butt there and just go, and then with your shotty, and it sucked through. Oh no, in there in the bowl, and that's your shotty. And this is, I was talking about Canada jobs last week and entrepreneurships in cannabis, and this is another cool thing with some cannabis art. And so you can even just put anything in, the grass clippings in it, and that's nice cannabis art, and you could sell them to cannabis people. Or this was for, I think this was actually proper cannabis, and that big one was 1/4 of an ounce. No, 7 grams. Hang on, that looks way more than 7 grams. Just thinking about that. That's nearly like 1/2 ounce. Maybe the small ones are, but still they're pretty impressive looking big fat joints. Look at, actually your mouth wouldn't even get onto that. You have to put a funnel onto it. This is another thing with cannabis jobs, entrepreneurships in cannabis. So you can make these types of charts up. So just pull the old school charts up and then put the new school information around it. And when I was going through, I thought, oh, this is pretty cool. Look, the CB1, CB2s. And look at down here. The tri-P, that's not a receptor. It's the TRPV1. Because remember I was telling you, your total real pain version 1, that's how you remember the actual receptor's name for the CBD. So, yeah, that's how shit it is. People don't even they go to all this effort and they can't even fact check stuff like that. So if you do it, just do it properly and you'll probably do a lot better than these people. Endocannabinoid. Yes, but it's good. Jobs. And here's another thing for these are grapes. They got heart-shaped grapes and star-shaped grapes now. And this is done through breeding. And it's more so in the selective breeding, not really in the marker breeding, because you're looking through morphogenic expression to see what shapes you're going to get. So you put mutagens into them and you would change the DNA to come out and get these different results. And then you just pheno-hunt and pick the ones. So that's how they did it, but I just really like it. It just shows there's so much more that we can do in plant science. And this is cool for jobs, for entrepreneurships and cannabis jobs. You can get there's people that brew all their kombucha or they brew alcohol and stuff too. You can add THC, so you can get tinctures which are very pure in it, and then get THC. And this is enriched with 10 milligrams of THC. And they said this is in California, and I just thought it's pretty cool. Just it's going to be in everything. I even saw a thing for because I'm doing post-harvest productions of crops, and you can even freeze dry ice cream. So you can infuse ice cream, freeze dry it, and it looks like trail mix. It's it's hard. All right. So this is to look at the results of high temperature on plant cells. Another study done to say that the transcription factors, the PIF4 and the TCP4, had a massive play in changing the pattern here of the stomata growth. You can see from the high temperatures, it makes the stomatas go. They're not as dense per square inch, or square centimeter, whatever it's measured in, and their cells. See the cells in between them? They're a lot bigger as well. So there's a big difference. So that's the high temperature. So that's photorespiration. When it gets too high, the temperature's too high where it can't photosynthesize properly, and rubisco is the enzyme, and that changes to, instead of CO2 as the input, it uses oxygen as the input, and it's still photosynthesized, but it's not as healthy. And that's what happens with high temperature. And this is a similar thing. This is the plant's growth patterns from water losses, like we were just talking about. So here is sufficient soiled water. So this is all sweets. This is what it should look like on the left. And over here is in drought conditions, or when the water's been lost from the cells and stuff. So see up here in that spongy mesophyll layer, palisade in there, they're all fat and juicy. And over here, look at, they're all shrunken. And in the xylem and the phloem, I can't tell much difference in that. Maybe the walls? No. Anyway, down here you can see it's also in between the cells. They're shrunken, so the turgor pressure is lost. Oh, here it is. It says on the side. Oh, shit, eh? All right, go back to the top. Turgor pressure is lost. Yeah, the cells. Stomata's closed. Oh, yep, you can see it shut down the bottom. And down here, there's reduced tension. Okay, we can't really visually see that. Fine roots and the lower turgor pressure. Yeah, you can see there's all the spaces in the middle of it. It's also its water flow. All right. This is pretty nice. This is a different one of trichomes again. So you can see, oh, yeah, it shows the different types of cyclic trichomes. There's your sessile, and then you got your capitate, and then you got your stalked capitate, and then your uniglandular hair is this one, and then multicellular. So you can see the cells and the cell wall in the middle. It's so nice, isn't it? It's really snazzy. And you can see the ambitrichs there, and these ones have been aged, so they would have been developed first, and they finish first, and that's what you want. That's when you know everything on your crop is finished, if it looks like that. That's really nice. So still no questions, eh? That's cool. At least I'm looking today. Molecular pathway. Oh yeah, the flowering locus gene. What's going on with this? The pathway to relocate the flowering locus expression to seed development from parental vernalization. Oh yeah, okay. So the parents got vernalized, which means they got put through a temperature process where you can you put it in cold temperatures, say in the fridge temperature. You germinate the seed, have the radical come out, and then you put it in the fridge, and then what it does is it initiates a strong flowering pathway. So there's a few ways you can initiate your florogen gene and your hormones and florogen, and this is one of the pathways through vernalization. It's also done in cold climates, so use up in North Canada and in those cold regions in the mountains. This is a technique that you can do also to induce flowering, strong flowering, and it actually helps in cold climates, vernalization. It's pretty cool. But back to it, the flowering locus gene got activated from going through its growth development stages. How though? Oh, because the parents got vernalized, and then in its progeny from the seed development, when it was grown, it's shown these traits. So it just proved that the progeny did pick up what the parents had, and you can have vernalization coding through that comes into its daughters and offspring. What? There's more than that. It's saying it's all that's left. No, I'm going to have to look. Stop sharing. Hang on for a sec. Oh, okay. I'll say g'day. Hello, Dan Boys Grows, Lene M. How you going? Sunshine. Ned Kelly. G'day. How you going, Jeff? Stony Creek. All right, Stony Creek. My chat's not working on my. I tried to pull it up on the other thing, and it's not working, so I couldn't see any of those messages. I'll try and muck around with it. I don't know how to get it going. Oh, it's not. Yeah, oh well, sorry. I'm trying. All right, back to it. This is another thing, was video file share screen. So this is the effects of potassium. They did a test to see what the effects of potassium are on plants, and it's pretty cool. This is done April, a few months ago. So they did they had these milligrams. So they fed it 15, 60, 100 milligrams, 175 and 240 milligrams at different intervals to see what the outcomes were, the results. All right, the results were, let's see if I can zoom in a bit here. No. Oh, fair income. Oh. I just lost everything. Sorry, guys. I'm supposed to go down and show me the results. I was going to zoom in to show you. Oh, I'm not going to zoom in this time. So you can see the difference in the buds. So it's 60 was the best. So you can see the size of the bud. 60 was the best, and that's what they found in the outcome. And you can see in the leaf here as well. I'm going to try and zoom in because you've got to see this. Please don't. Hey, there we go. So you can see in the leaves here. So that was a little bit deficient in potassium, and then these had toxicity in potassium. And that one's perfect. So 60 was the outcome. So that's all I really want to go and say with that. All right. And there was another thing. I'm sure there's another thing. How you going, PTP? There was definitely another thing. I'm going to go back look in the thing. I'll share because I'll show you what I was supposed to do today. Entire screen. This is all the stuff in here today. There was something else, I'm sure. It was for Colombia. Photosynthesis. Where's the... Oh, I didn't put it in here. There was one about the stomatal conductance and leaf potent. All right. Stomatal conductance. Oh, that's it there. I didn't put it in. Come on. I want you to see all my stuff. This one here. Why isn't it working? Oh, sorry. I'll just save it for next week. Stuff it. Well, that's about all I got to say. Is there any questions, anybody? Anyone? I'll scroll back up to the top, see if there's any, because otherwise I think I'm going to go. I'll put the invite thing, see if anyone wants to come in. That's in. Now scroll down. Howdy. What did I miss? I didn't miss much. You can go back and see. Good to see morning. All right. This is the show. Okay. That's it. All right. Thanks everybody for watching and joining. Save your questions for next week. because this week's just is finished. I got nothing else to talk about. Oh, hang on. I know you talked about this before, but what's your method against botrytis? Huda 420. Botrytis, well, I'd go beneficials. Bacillus subtilis is really good. So I do IPM when I flip, and that allows 2 months worth of protection. So I'll make up a nice brew and make sure I have Bacillus subtilis in it, because I've had botrytis before. It sucks getting that scenario. It's not very good, but it's a humidity thing, to be honest. If your humidity in late flower gets above 65%, it's actually at 60 now, the water activity level. That's, can you hear me? Make sure I'm blooming. Can you, yeah. If your water activity level gets above 60%, that's good for aspergillus, but that's not, it goes green and stuff, but that's not full on. But above 65 is definitely when I'll shit me pants. And especially if you've got big chunky ass buds, it might be 65 in the air, but what about in the center of the bud? It's higher again, and that's where it grows. So good soil health is to have heaps of pathogens as well as good microbes. So the chances of, if you put it into those conditions, or if things lessen, if the environment slips or the plant's health lessen, that's when these things proliferate, because everything's always around. So I've also heard from milk. You can spray milk. Labs works, but this is in flower, so I don't spray flower. So yeah, if I was to get it in late flower, I'd chop it down, because there's nothing you can do. You have to just cut it out. You can't even extract it, because you get all the mycotoxins and stuff in it, and it's not healthy. They sporulate. Outdoor field grown, you can't control though. Outdoor field. Well, you've got to do your IPM. You've got to get your plant health up. Make sure you do soil tests to make sure everything's in the soil that's correct, and you've got good aeration, because you don't want any fungal growth in there. And you can get genetic resistance in. I've put genetic resistance into my line. That's a good suggestion for if you're going into outdoor fungal growth areas that are high in humidity and late flower. You can also put get big tents or little hoop houses and put that over the top, and put a dehumidifier or fans extractions inside of it with some meters. Yeah, and then thin it out. If you're knowing the humidity, for instance, in the Pacific Northwest, while I was over there outdoors, you have to go at day 40, probably day 40 odd. You thin the shit out of it on the inside. You want to see through the plants from one side to the other, and that's going to really reduce the chances of the humidity spiking above the 65, or I think it's about 75 botrytis, or 80. So but it's above 65 is just the lethal level for me that I watch out for. Yeah, that's about what I'd do for the outdoors. Genetic resistance. So if you're going to get that every year and you can't combat it, genetic resistance. Get someone like me who you can put those codes into your genes, and you can make it resistant to that. I've actually watched my video on that. I did that a few weeks ago, released that one on fungal resistant genetics and how to do it and how to install it, and the systemic acquired resistance that you need and the induced required resistance to start it. Oh yeah, there's still, I should be looking at questions. Sorry. Oh shit, things just out. Yeah, is Nexus make an appearance? I don't know, mate. Sorry, sunshine. Is there a specific terpene that fights caterpillars? Yes. You want rhodonone is really bad. That comes in, I think it's skunkweed. You know, in the Pacific Northwest up there, there's the skunk. Oh, you're from Australia. Huda. Rhodonone is one. And caterpillars, you want, what's that thing that flies? It's called lacewings. Lacewings are really good for caterpillars. I'd get some of them if I was outdoors. Caterpillars, yeah, you can pick caterpillars off. That's the good thing about them. They're really big, but still they're going to infect stuff. So lacewings will work for that. What's Danny say? Right varieties for your climate. Yes, can't grow your strain where you live, just like other crops. Yes, you've got to adapt to your environment. That's what Stoney's saying. He's right. You do. And that's when, if you get cultivars that aren't ready for that environment, that's when they'll get all these problems. So you have to install the right codes in the genes to allow that to happen. So you can adapt it. So that's, you can do it through epigenetics, or you can do it quicker through a systemic acquired resistance. Yeah. Hey, Grommies. Kincaid, Cassius Clay. Hello, hello. Huda says, day 40 at flower. Would you defoliate after? Well, there's two reasons why you would and why you wouldn't. It releases ethylene gas when your leaves are going through senescence, so that speeds up your crop to finish, because ethylene, that's one of its cycles in its cascade signaling pathway. It goes through. Ethylene induces, that's how you can get an earlier finishing crop. You put more ethylene in there and it speeds it up. So if I wanted the crop, to answer your question, if I wanted the crop, I should show it. Oh, yeah, that's a good idea. And then you know what I'm talking about. Oh, cool. I'm not very good with computers. I'm really good with plants though. Fuck. How'd you defoliate after day 40 of flower? Would you defoliate? Only if you wanted to crop the speed up. To slow down, you could get rid of all of the leaves, all of the non-sugar leaves, and then that would slow it down a bit, so it would finish later. But I always tend to finish day 60, 65. So for me, I like to defoliate, because I like to let it finish naturally. I don't want to speed it up. If I was going to speed it up, if I wanted to finish early, I would let them all just go dry on the plant and brown, and then take them off then. And that means that it's already released its gas and it's finishing senescence fully, and it's already, necrosis has kicked in hardcore and it's died. I hope that answers that question. How do you put the codes in the genes? The easiest way is through induced acquired resistance, IAR. And that's kind of like COVID thing, where you give it a little bit of COVID to the human, and then inoculates and let the own immune system kick in. You do the same with the plant. You give a little bit to the plant of the problem, and then that kicks in. And then that, like at the moment, actually, yes. So you just inoculate it with a pathogen. The plant's immune system kicks in, and if the plant's in good health, which it should be before you do this, the plant will, about 2 to 3 weeks it'll last. So it won't get any growth, but it'll go back into its immune system kicking in phase, where it's got to try and fix up itself. So yeah, you won't get any growth, but it should fix up itself. And then you've got a pathogenic a prime gene. It's PR prime genes, pathogenic resistant genes. And that means that once it gets an infection next time, you've got PR genes primed and ready. So they just, bang, they're in action. Go, go, quick, send all the troops out. And it just gets in and hoses everything out, puts it down, spots out the fire type thing. That's the easiest way to do it if you're not in a lab, because otherwise in the lab you've got to use chemicals and you use different machines and stuff. But that's a way where the average grower can do it to themselves. What's pure protein? Some guys in Humboldt talk about it. Pure protein. Well, protein's amino acids, so they just say it's high in amino acids. Are pure? I don't know. There you go. Don't know. But amino acids bloody help, because they've got all of the right enzymes and codes in them to get the action. So once the RNA gets coded, it puts out to the proteins, and then that codes out gets the action happening, and then the proteins break down there and amino acids and stuff that is in them. So pure proteins, well, having anything pure, it's not really the best, because you want to dilute everything, because it sort of gets into a toxicity range. But I don't know. That's a good one, mate. I'd like to hear more about that one. Good question, Sunshine. Do you defoliate according to the moon cycle? No, I don't do that sort of stuff. The moon has a little bit of gravitational pull, and it affects Sheila's. Sorry, girls, but with your cycles and that. But no, I really don't. And the amount of turgor pressure, the osmotic pressure that's inside of a plant, is so low. It's if you compare it to the atmosphere, it's only like it has to equalize with the atmosphere, and then it won't have any transpiration. So once the pressure goes lower in the atmosphere, it has a suction pressure, and that's the cohesion tension theory that works to get the water going from the roots through to the out through the stomata, and that's how it cycles. So no, I don't do the moon thing, mate. Doesn't mean to say you can't, but I don't. Is it possible? All these questions, good on you guys. This is great. Is it possible to have a deficiency even if your leaf analysis reads good? Oh yes, it is. Great question. Example: iron levels reads good level, but it seems like there's iron deficiency in the plants. Yes, it's called a hidden hunger. Let me pull up a chart. Hidden hunger. I'm going to go into soil science. Actually, no, I'll just go into here. These are the classes that are courses that I've done. This is one of my latest little gems. Hidden hunger. So where was that? Hidden hunger. There's a good chart I've got. There it is. Wow. Hidden hunger. So you want to try and feed in these optimum ranges in here. And then here is your hidden hunger bit. So you're not really going to see any deficiencies, but it's a little bit hungry. Meaning that if you increase your yield a smidgen from this bit, and you increase your nutrient uptake from this position when the plant's in this position, you'll get a smidgen more yield. So that's what old mate's talking about. Sorry, I haven't got your name. Hidden hunger. Yeah, it's happened. Same with toxicity. You can go over hidden. So when it's all the leaves are looking fine and stuff like that, there's no types of any symptoms, you can just go a little bit too over. So that's why people always say try and go a little bit under when you're feeding, if anything. And next week I'm going to be doing a nanotechnology in agriculture discussion, and that's going to go on about all of those types of stuff. What else we got for the hidden hunger? This looks like it's around that section. Yeah, your critical range is where you want to be. Sufficient. Oh, critical range. Nutrient commission is the plant which it yields responses occurs in natural. Yeah, sufficient, optimal. Chelation. No, we don't want to talk about that. Back to the question. How to identify deficiency in the soil? Nope. All right, that's it. Do I stop sharing? Yes, I did. Cool. Thank you for your question. I hope that answered that one, mate. What would the pH be affecting the nutrients deficient delivery, even though the leaf analysis reads good? So how would the pH affect the nutrient delivery if the pH reads good? Oh no, I didn't share that screen, did I? I just went and explained all that shit, and then I didn't share screen. I found what I was looking for with the hidden hunger in the chart. I'm sorry. I'm going to press share screen now again. Who to 420? Good question, mate. I'll show you the answer to your question. Oh, so on the screen, there's the question. Wow. It's hard to do admin at the same time. Allow. So now you can see everything that you're looking at. pH. There it is. Righto. So even if your leaf shows fine, everything's fine. If your pH is just starting to go out, your leaves will start to show symptoms, and this is the reason why. Because if you're running, they always say run 5.5, 5.8. No, I don't reckon. Because look, molybdenum, you're virtually locked out of molybdenum and CalMag. So everyone says, oh, what about CalMag? And phosphorus. So once you go up above 6, it starts to get a bit better. So I try and stay around, I used to do 6.2, but I do about 6.4, 6.5 now. And that gets me availability a lot better in this range. So if you even stick about 6, you're still going to have a CalMag and a phosphorus potential lockout deficiency. Nothing you can do. Everything's going fine. You're feeding it right. Everything's going right. It's just a pH issue. This is why you should always check your pH runoff. If you have runoff, check your pH. If you don't, try and get a soil sample and try and stick it in a cup and try and do a pH test. And If you've got problems, this is the first thing you should be using to diagnose, because more than likely it's be a soil pH. Then you want to just go and lime it up if it's too low, or gypsum if it's too high. So you put a bit of calcium if it's too high, because that's going to bind things up to extract all of the bad things, to reduce it, and the same with to raise it up. You go and lime it. There you go. That's that question. Good question, mate. Thank you. Who was that from? That was from Huda, 420. All right. Some of my Kincaid Cassius Clay, some of my baby leaves have black tips. Is this due to overwatering? And one has yellow tips. Is this due to nutrient burn? Well, yellow tips, yes, but you'll usually see the yellow tips. They won't be on your new growth. They'll be on all your growth. And that's the sign of when you've got saline soil, means that your salinity level has gone too high. And if you did a test on that test, you run off with a ppm meter, you'd see that your ppm would be above 2000. And that means that your plant has, even if your pH and your nutrient availability is fine, your plant still can't get access to that, because it can't, it's a hypertonic solution, which means that there's too much salts in it and it can't exchange. So you have to flush it out. So if you ever get the ppm too high, just flush it out, and then put a little, then redose it with a dose of, you know, your NPs or whatever you want to give it at that time, whatever stage you're at. I didn't put the thing on the question again. Would you, no, someone, it was this one. There you go, just to show the question. Some of my babies have black tips. Oh yeah, some of my babies have black tips due to overwatering. The black tips. Well, that might be, if you've got any, it's only black tips. Well, something's going on. That's good on you, mate, for reading the leaves, because that's the first place it's going to, you know, really indicate something. So good on you for that. And black tips, that could, well, usually with fungus. When you've got a substrate problem, you get a fungal problem, and that's curves the leaves and canoes the leaves. And I don't know about black tipping. Black tip. Have you got any stem cankers? You might have something on your stem. I'd look under your leaves for that. And if it starts to increase up if it starts to get necrotic leaf, the necrotic cells are going to grow up your leaves. In other words, is the black bit's going to get bigger. That means you've got a substrate issue and you've got a fungal infection. So beneficials, pump the beneficials. Reduce your water. Make sure you've amount of, next time you put more perlite or grow stones or something to aerate your soil a bit better, unless it's outdoors. I hope that's helped enough. Fill me in if it's hasn't. Next question. So you would inoculate botrytis into the plant. How would you actually inoculate? Well, when I've done it, I've put it in through the substrate. You get the spores and you'll put it into a blend and just feed it to the plant. It's so bad. And then I got some cool photos, and then allelopathy happens. That means it can speak to the other plants. And if it's within, I'm not going to tell you that because that's pretty cool stuff, but they will transcribe the other messages. It turns the signal into a methyl signal, methyl signal, and then that goes airborne and volatile, and then it picks up by other plants' receptors, and then they change it from methyl back to the normal, and then that is a jasmonic response, jasmonic acid response, and it induces the resistance into the plant. And then you go and do tests afterwards, and then you'll do a pheno hunt. So you'll have your strain growing up, your cultivar that you've just made, that you put in with your pathogen, and then you'll create progeny from that, and then you'll do a pheno hunt from that. So after you breed with her, you will pheno hunt, but with this pheno hunt, you'll be putting it in a conditions that is in like 90% humidity, and then seeing what happens. That's it. And then that's what I did in the video. And then you can see what happens. And then you go, wow, look at you. You've gone and you've got resistance. You've gone and only seeing growth on dead cells, not live cells. In other words, it can withstand fully withstand the disease. That's how to do it, mate. Good question. Good question. Huda 420. Yeah, probably aminos. Chart isn't showing food at 420. Sorry, mate. Jesus Christ, I'm bloody. I should nearly pull it back up. If there's no more questions, I'll pull it back up for you. Hello, buddy. Huda. No, can you please show Hidden Hunter again? Are you where Steve Solomon's work? No. Do you agree with his soil targets? Don't know him. Is he pushing nutrients too high? Well, if he's pushing nutrients, if his level is getting into the sodic level, into the saline level, is 100% too high. It's physics. It can't work unless you get salt. They're called halophytes, when that means that they're salt resistant or they're salt tolerant plants. So unless you put halophytic genetic genes into them, which you can, I don't suggest doing that. I've seen people. My friend, he used to feed the plant 2EC, which is like nearly saline, nearly at the level where you don't want to do it, but he gets mad results. So it depends how you set up your substrate, because remember, for good soil health you need three things. There's nutrients. I'll show you the chart because it's pretty cool. Actually, I'll go back to Hidden Hunger to show old mate first. Share screen. And then I've already forgotten what I was talking about because I wanted to help him with his Hidden Hunger. This is the chart again. Not again. This is the chart I was supposed to show. So in the middle is optimum. Outside is your Hidden Hunger. So really we're talking about in here. So you can improve your nutrient uptake, but you still get the same conditions where it doesn't show any symptoms on the leaves. Hidden Hunger. I spoke about it before. I don't want to sort of elaborate again. What was the other thing I was going to show you? Is he pushing limits too high? Was I going to show you the sodic stuff? Charts on that? I can't remember. Sorry. All right. So botrytis, is he pushing nutrient limits too high? I hope I help. So botrytis I'll show it, can come up through the roots and show symptoms. No. Well, it's systemic, so it'll get in through in the phloem and the xylem, but it doesn't really do it that way. It's spores. It's how it happens to the plant is spores will fly in, they'll get and land on the plant, or they'll be already on the leaves from the air, because there's spores everywhere. You know, there's so many thousands around us, billions. 1 gram a teaspoon has got billions of bacteria and microbes in it. That's how the plant gets it. Then it's got to germinate the right water activity level, so it's got to be above, for botrytis, it'll have to be above, I don't know, 75, I think. And to give the resistance is you want to work each cells. So the cell is going to code up and induce the resistance into the cell. That's going to write the RNA signal and transpose it to the neighboring cells, and then it's going to transport that through the system of the plant, through systemically, and then it should be throughout the plant and have a systemic resistance. That's what I meant by it comes up through the roots. That's how I'd feed it that way. But botrytis doesn't form through the roots. You get botrytis is it's grey mold, it's on the top, botrytis cinerea. For the roots is dampening off, and that's Pythium, and that goes black. That's the one that you'll see. That's another fungal infection. That's bad. I've worked with Pythium the same way that we've just talked about. Cassius Clay, thank you. You're welcome, mate. And the baby with yellow. Oh yeah, that's a good one. I didn't finish answering. Sorry. And the baby with yellow sprouted yellow leaves. What does that mean? Well, if it's a mobile element, it'll show in the older leaves because it can be transported from its source cell to its sink cell. That's how the plants work. And if it's a non-mobile element, meaning it can't be transported around, translocated around through the plant, will happen. You'll see the deficiencies in the new growth, and that's The non-mobile ones are calcium, boron, and manganese. So that's where you'll get, if you see a twisting of the leaf or a bit of even a yellowing, like you suggested, it could be calcium. There's not many questions left here. I'll go and pull up a slide and show you because it's fucking rad. The share screen. This soil science class is fascinating, guys. It just put me into a really good perspective on how things go, how it works. I didn't even know I was going to show you now. Oh, damn it. I forgot what I was going to show you. I just love this course. I'm going to have to stop sharing. I don't even know what it was. Thank you. Oh, that's right. It was the mobile elements. All right, back to it. I need an assistant. Where's Dr. Nexus? Mobility and elements. It was right at the start. Oh, that was a rad thing. Here you go. This is this. Oh, that's too complicated for now. That's really good about the cation exchange. Asking us the cation exchange questions. There's calcium. So this is the calcium. So we'll go to the tissue. This is what it shows in the that's where in the early growth. Different forms, how to give it to it. That's the different forms you can get it. If you want exchangeable though, so the plant can uptake it. Deficiency. This is what I was talking about. Here we go. See if I was right. Deformation of the dying tips, meristems, necrotic leaves, curling, stunted growth, black spots in the leaves. Hey, look, there you go. Oh, but it doesn't say leaf tips. Yeah, black spot. That's a bit different. In mild deficiency. I really like this one because it has to be twisted. And some of these photos, they don't show twists in them. And there's a little curling. There's your black tips. Tip burn. Oh, yeah, actually, there you go. You got a calcium deficiency. If it's yellowing and black tip burn, there you go, mate. Sweet. All right. That's helped you. Now I'm going to go and show mobility. There it is. No, it's not. Come on. There it is. So the mobile elements with plants, this is inside of the plants because remember if it's in the soil, it's not mobile because it's hard to get around the soil. So you got nitrogen, phosphorus, potassium, magnesium and iron. They're all mobile. And immobile, it's calcium, boron and manganese. And then they made a new section on this 2022. The old section that just used to have immobile and mobile. Now they've got variable mobility, and that's copper, zinc, sulfur, molybdenum. That's pretty cool. These are some other charts. I'm going to show you this one too. This is the plants can only uptake nitrogen in two forms. So it's your nitrate and it's your ammonium. So if you've got all your other forms, they can't accept it. So when people say, why can't you give it the right one? And that's why some fertilizers, some synthetics, they won't have these forms. They'll give it to you in a form that's not acceptable for the plant, and then it'll have to be broken down by microbes for the uptake of the plant. So the good nutrients, the good ones, have these available. They only have nitrate and ammonium for a nitrogen fertilizer. But organics is the go, and you want to try and unlock it. And I don't want to go too much of a spiel, but these are the actual ones that you need for a plant. So you want nitrate, ammonium, your orthophosphates, so it's your primary and your secondary phosphorus, calcium, potassium's just as it is, calcium's like it is, same with magnesium, sulfate. Then you got your two forms of boron, is boric acid and borate. Then for your copper, is cuprous, is the only form that's for plants accept. Chlorine, it wants chloride, and for your iron, it wants ferrous. Manganese is manganese, and molybdenum, it gets it in the form of molybdate. They're the ones that it wants. Anything else, go away. Next question, or anyone else needs to be unlocked, actually is the better word. Good question, mate. Huda, wounds on flowers. Yeah, well, if you wound your flowers, that's not the... G'day, Monty, how you going, mate? Wounds on flowers, it will induce problems in the plants. It'll think, shit, I'm getting attacked here, so it starts to not put its growth out, and starts to activate its immune system. So that's why all those shock treatments, they do maybe have a little bit improvement on secondary metabolites, but it limits your growth, it limits your yield. So there's better ways to get improvements on secondary metabolites than to wound your flowers, I suggest. That's a good Educator Monty. Good question though, Huda. All right, scrolling down. Oh, there's another one. Hey, Main Green, how you going, mate? Is it better to have high deficiency of nutrients with correct ratios or a lower deficiency of nutrients? Shit, eh? It's best to have a lower deficiency, because if it's too high, it'll be in the saline condition where there's too many salts and the plant won't be able to exchange the nutrients to get what it wants. So it's just 100% locked out. So if anything, it's got to be lower. And then some nutrients do react with other nutrients. And that's getting into a bit more technical. I'd have to show slides to really discuss that sort of stuff to be accurate. But yeah, stick on the lower side if anything. You want to make the plant work for itself. If you baby feed it, it's going to sit there. Remember, like the chickens that are in the cages, they're just sitting, you put them out in the open, they'll fall over. So that's what you do with synthetics. You're just getting a yield only, where you want the plant to work for itself and improve the secondary metabolites. You're still going to get a good secondary metabolite profile with synthetics because they've got the formulas down pat now, but you can get better ones. And you want the plant to work for itself, and that way, when it works for itself, it has an immune system activation where it can defend itself. If you baby feed it, it's just I want pesticides to feed this disease. Oh shit, I need this to feed this to combat this section now. Where if you get the plant to work for itself, it naturally does that. Like us as humans, we naturally have our defenses. The plant's the same. They've got the same eukaryotic cells, and it works a similar way. Good question, Huda. Cheers, Main Green. Where's the best soil in Australia to grow cannabis, or the best region? The rest, soil in Australia, you make your own. Soil's very futile. Remember, it needs three things. I'm going to show you now. You've got a good soil question up. Let's see why does it take good soil? So you need your good chemicals. You need good nutrients. What's the third thing? And you need good physical characteristics of the soil. And to get all those ones equals, you want to be in the right bracket, and that's what the plants love. So we're only just talking about this the other day, so that's why I can skim to it. I went through a fair bit of soil testing too, guys. I'm getting some really good soil testing stuff. Soil testing, then you know what's up, then you can know how to act. So what was I talking about? Oh shit! Quick. Soil, something about soil. Oh, that's soil health, rad. Here we go. So soil health is the continued capacity of soil to function as vital living organism that sustains plants, animals, and humans. So you want to have three things: biological, chemical, and physical. And if those three things are in check, you end up in this blue bit in the center, which equals good soil health. So if you've only got chemical and physical, like I had when I go to the paddock and I go and get some grass, which is heavily in clay, it's got very low microbial content, you're in this level. So that's the level to win anyway. I think I'm talking too much. Good soil health is a relationship between soil health, its functions, and the indicators. So you've got to make sure you get all of these things in check to have good soil health. Actually, I think I'm getting off the point. Yeah. Yes, so the point is, make your own soil. You get heaps of organic matter. You want an organic inputs very high. If they're above 1% soil organic carbon tests, that's considered really good substrate. It has a very high cation exchange, meaning that it can absorb and hold on and chelate a lot of beneficial nutrients and elements and ions that the plant needs. So if you haven't got that, it just gets washed away and it just sits in limbo and sand. What's it doing? I don't know. It just gets washed away. So you need to hold on to stuff. That's what the organic matter does. So make your own. You make your own with vermicastings. And there's a real easy way. It's called the Indore method, INDORE. It was developed and you use cow dung and green manure, which is all of your leaves and shrubbery, bushes, shit like that, without disease in it. And you put your cow manure, which has no antibiotics and no steroids and stimulants into it, and you put that together. You put a little half moon shape on the ground, no more than 2 feet thick, because otherwise it'll go anaerobic and grow all these mycetes that we don't want, like pythium and botrytis and shit. Yeah. And you make your own. You get the worms to come out of the ground and do your own for you. Then that'll turn over. You turn that over 3 weeks, 3 times, and then it's fixed. You seal it off and then you seal it back up with slurry made from cow dung, and that locks in the nitrogen, because otherwise nitrogen fixing bacteria, they will exchange it and they'll transform. It'll go back into the dinitrogen, which means that the nitrogen gets formed back to volatiles into the air. You want to try and retain that, so put a cap over the top, and that locks it in when it goes through its composting phase. That'll do. Aussie Grow Show, cheers. How you going, mate? What's your germination process, says Hooter. Well, germination, you put it in water. It takes 3 things for a seed to germinate: temperature, oxygen, and what's the third thing? It's temperature and oxygen. It's the environment. There's temperature, oxygen. I thought it was 3. Anyway, so I just put it in the water, leave it on the top, and remember, water exchanges oxygen 10,000 times slower than the air. So I used to push my seeds down after a day and let them sit at the bottom. I don't do that anymore. I let them sit on top and let the radicals come out. And once it gets to about 1/4 of an inch, or what's that, 3 mil, I'll take it out and then I'll germinate it and go from there. You can do hard breaking ones. If I've got ones that are a bit hard, I might put a little bit of ethylene gas into it and just put in an apple into it as well, an apple core that will release the gas, and it induces its pathway for germinating. You need gibberellic acid and ethylene to stimulate its germination pathway for a seed. And yeah, that's how I sprout it. You don't want to go into dormancy. That's the germination process that I do. The one with black tips under the leaf is pretty much completely black. Oh, the one with black tips under the leaf is pretty much black under the leaf. Okay, well, you want to expect that a hell of a lot more, because you've got something growing onto it. And if you've got black stuff under the leaf, it's downy mildew. Powdery mildew grows on the top and downy mildew underneath. So that's the type of fungus that you've got, if that's the case. And yeah, get back to me if that's true. I hope not. If it's the case, you want to reduce your fan content, lower the humidity, get rid of those leaves. Remember that they're spores, and one spore, it produces dust. It's like, that's how they get round. So you have to be so careful, nearly even water them down before, like, do a spray with water and then pluck the leaf off so the spores don't get airborne, and get yourself a dehumidifier. Poor bloke. What's the best soil test company in Australia? Good question, mate. It's a fucking niche market. It's if you want to pay, if you've got thousands of dollars, you'll be right. But if you're like me and piss poor, it's a niche market. It's something that I actually looked at creating. There's it's called a soil health card, and I was going to get onto the government and look into it, and people will give me their samples, and I'll do the 5 tests that you do, and then send it back to them, and with fertilizer suggestions or ways that they can improve their soil to get it back to the condition that it should be for the plants to love, the organic way. That's something I looked at because I can't see it available, to be honest. I've been looking for testing stuff for quite some time. I think it's only if you've got a commercial license, you can put your ABN to it and pay these labs probably a couple of grand, and they'll do it. But I don't know, mate. I haven't found anything. I'd like to see something that's affordable for us people, because we're the ones that are really teaching these pricks, these big fellas. Look, they're all going in the bust. Remember, the first company for Australia has already gone into liquidation for medical cannabis. Good question, Hooter. Look at high-powered organics. Huda, it says sunshine. Has indoor soil proven to be more beneficial to growth? And how can soil mix help indoors? Has indoor soil proven to be more beneficial to growth? No. Soil is soil, and it's how you make it up. So each cultivar needs a different requirement for it. Like I've got pineapple is a potassium pig. My old Nukin in BC used to be a nitrogen pig. So you've got to amend your soil to your cultivar, and then you'll be right. As long as and get your organic carbon up high, because it's all about that. It's the plants, they sit there for months on end, and if you've got them in a pot, you've got to try and keep that healthy for that duration for months, and those microbes and everything factoring in, and try and create your own little soil food web somehow. So I use 25% worm castings, 25% organic compost, and 50% perlite. That goes into my organic substrate. And then you'll see little worms coming out sort of, I don't know, day 30, 35, they'll be coming out because things are getting too dry, and the amount of carbon and everything in there that they require to stay for their requirements isn't suitable, so they just want to bail. How can indoor soil mix help outdoor? How can indoor soil mix help outdoor? How can a indoor soil mix help an outdoor soil mix? If you want to reuse your soil, I suggest do it, because it's soil. It's not dirt. It's not inert. You just got to recharge it and get it back to its potential that it should be. So reuse it. Stick it outside in your veggie garden, and recharge it again. It might take, you'll have to cycle nutrients through it and different, compost it. Tip it into a compost heap and leave it for 12 months. And then you don't do nothing. That's probably an easy way. It's really good to reuse your substrates in a good way. But remember, if you've got problems, like old mate before was talking about, he's got his fungal substrate problem, then you don't want to reuse it, because your soil health has got an imbalance, and to fix it back up again, it'll take time. So yeah, even though I do, because I like to work with problems, because it creates genetic strength, but it's really hard sometimes too. So I wouldn't sort of advise that way. I hope that helps, Aussie Grow Show. Test company. I know you're looking around at 2K for soil analysis. For example, bringing similar mediums outdoors. How is bringing simple mediums outdoors, has it show any different in performance? If you by using an indoor substrate outdoors, well, if you use like a ready mix from the hydroponics shop outdoors, well, you can do that, but I just make up what the plant wants. Just give it some compost and worm castings, because worm castings, they've got all the minerals in it that have already been cycled, and the compost has got all the stuff that's ready to be broken down by the bacteria and fungi that and the archaea that's already in the protozoa that are in the compost, in the worm castings. So you mix the two together, and they cycle through for the next 60 days and release all the right things for the plant that the plant can uptake. So I would I just make my own soil. Now I know a fair bit more about soil. It gives you a buffer zone, where if you're using like cocoa, for instance, you've got no buffer, where things have to be done very fast and problems happen super fast. Where with soil, you've got that buffer zone where you can just allow, whatever, but it happens, and things happen a bit slower. Hope that helps, mate. Is tissue culture the future of the cannabis industry? Which media is best? Which MS media? I use a modified MS media that has extra sucrose and vitamins and minerals put in it. So then all I've got to supply is my hormones. And the the gel. And if you're going to, it depends what type of tissue culture you're going to use, if you need a gel or if you're just going to use a suspension culture, just with a normal BMS media. And it is the future. It is fun as hell. I can only do it in summer because you have to create lab type conditions. So to do that, you want to be it always has to be at the stable conditions for it to go well. Otherwise, it just doesn't go as well. You get a lot of problems. You get fungus. You want to replicate plant cells. They divide every day. Every 16 to 20 hours, they'll divide. Where a bacteria or fungal cell, they'll divide in hours or minutes. So if your conditions aren't prestige, you're growing bacteria and fungi more than you're growing plant cells. So it can that's why I wait for the summer times to do tissue culture. But it's fun as and very easy, and I really suggest anybody to do it. And actually, most of you guys and girls have already done tissue culture. You cloned before and put roots out, bang, there you go. You have successfully tissue cultured a cannabis plant. Well done. So I suggest look into the Murasagi and Scrooge medium with vitamins and minerals and sucrose or fructose added, and then you're pretty much there. You don't have to spend top dollar on these damn kits and then half these kits don't have the right stuff in them. It's all about sterility, being sterile and having the right 5 things the plant cell needs to replicate itself. Good question, Huda. I'm enjoying this. Thanks for these questions, guys. Monty, I don't bother putting them in a cup or water until they sink. I just use paper towel method. Yes, paper towel method's very good for germinating seeds. Powered organics. So many soil testing companies in Australia. What are they, Monty? That's what old mate was looking for. Some universities in the US offer free analysis. Oh, wow. That's cool, the American one. Wow, that's rad. I'd be there all day, altering this, altering that, just to see what the outcomes would be. That's sweet. I'm just scrolling down here. Cost a few hundred to test them, and then a few thousand to rectify the issue. Oh, organic matter. Anyway, yeah. So thanks for that, Monty. Oh, another question. Do you know much about the Thai industry? Are they growing outdoor organic? Do they have good genetics? Are they doing things differently? Well, the Thai industry, from what I see, it's very, very early in their stages. Like I even contemplated, thought, wow, I wonder if there's good opportunity over there. It's too early in its stages. Like, if you're a bong smoker, I think you'd highly succeed in Thailand as a professional cannabis grower, because they really don't. From what I've seen, there's obviously companies doing it and people in the know, but there's not many. And because it's been given an open slate now, and the cannabis plants have been supplied by the government to the population, everyone wants to do it, but they just don't know how. So there's good money to be made over there, yeah. And Thailand know that. That's why they've unleashed this to the government, to their population. It's about all I know in Thailand. Sorry, mate. Compost and worm castings is easy, effective. Yes, sunshine. Costs you about $50 to set up worms. Yep, it's well worth it. Well worth it. These organic farming can nearly be sustained by only having maybe 4 cows, and it depends what sort of crop you want to put out. But if you've got one person, you can have 4 cows, and that's going to easily give you enough fertilizer for your whole crop. So you're going to have no inputs at all. You're only going to have outputs from your farm. You'll be taking out produce, taking out shit, taking out milk. You won't have any inputs. That's the future. I have a property and get the soil tested. Has been a big want. So, okay. Okay. Oh, here we go. Do you know about algae? Yes. Would it be worth getting algae to soil on purpose? This is a great question. You're getting all my fucking secrets, I tell you. This is an experiment I'm playing around with at the moment. I've got a He's watching me or something. I'm using Athrospiroplatensis. It's an algae that's very beneficial, and it's a nitrogen source, mate. Yeah, they're green. They got chlorophyll. Chlorophyll has a magnesium atom in the center with heaps of nitrogen around it. So that's exactly what you want. So you're onto it, Hooter. These questions you asked suggest to me that you're a connoisseur. Good work, mate. Keep up your good work. You're going to get rad stuff if you haven't already got rad stuff. Yes, and a lot of people grow in black pots for this reason, to reduce the algae. Well, I've been doing the opposite. I've been growing in clear and putting in different species of algae to see the outcomes that I can get. And I'm impressed. I'm still in the middle of the experiment. So you've sucked that one out of me pretty well, Hooter. It works, mate, but not with black pots. Beneficial algae breaks down to like spirulina is the type of algae, and that's 3/4 of the spirulina is what's it called? Tryptophan. Tryptophan is a precursor for auxin. Auxin's the rooting hormone, which is really good for roots and organogenesis to produce shoots. So by having spirulina growing in your substrate or even dried, when it breaks down, that's the chemical that it releases. So the plant can use this tryptophan, and it doesn't have to supply tryptophan to itself. It can like skip that phase and just go straight to the auxin, because remember, everything's a light switch. So it's got to switch the light switches on to go to the next step. So if it's got something where it doesn't have to, like this tryptophan, it can skip it and go straight to auxin, and that's the outcome. Yeah, that'll do. Can't give you too much. Same, been getting shit. What? Been getting shit results from a veggie patch and finally did what's needed. Ah, good, you fixed it up. Aerated teas and booster microbes. Yep, because they're going to stabilize things and get it back to where it wants. And the pH is the first thing you check. So if you haven't got the pH, because the microbes, they can't function very good below 6 pH. So that's the first thing you check. It's good on your sunshine. He's onto it. Sorry, but can you list exactly what ingredients used for tissue culture? No, because this has already gone on a bit today, but I can have a tissue culture. What do you call it? Like next week, I was going to talk on nanotechnology and agriculture. Maybe the week after, I can talk on tissue culture if you want, because it's a bit of detail. I'd have to show you slides so you get the right thing, because there's a lot of shit out there. Like, there's so much crap. Everyone's trying to capitalize in this cannabis industry, and there's a lot of shit out there. Probably 7/8 of the stuff, or no, 3/4 of the stuff is just, I don't know. Too much marketing goes on without the testing and the proving of it. So, Murasagi and Scrooge medium enriched with your vitamins and minerals and sucrose. That's a good start. Source that. I had to source mine from Portugal. So just source that first. And then by the time you hit that, you can ask some other questions. I get all scraps from work and convert. Oh, well done, sunshine. Can you go further into silica? Is there kind of different kinds of silica? And how can you make it most available to plants? Silica is really good for the cell walls, strengthening them, and for resistances for pathogens and abiotic stresses. I get mine from langbonite. It has 20% silica in it. I'll have to show you slides. My hands are a bit sore. I'm disabled in the hands, guys. You probably don't know. I'll have to show you another time. Can you go into further detail? I did an episode a few weeks ago on silica where I talked a bit about that. Different kinds. Well, silica dioxide. Where is it? Jesus. I'll try and find. Actually, my hand's a bit sore. I'll come back to that if there's no other questions. Hooter? because I sort of had answered that a couple of weeks ago. Is neem oil safe on seedlings, or do they have to be a certain age? Neem oil is really, really good. It's active ingredients azadirachtin. It's an anti-feedant, and it works really, really well against repelling a shitload of insects and bugs and microbes and everything. I'd dilute it if I was going to put it on little ones. You can do it. I'd do a test. So with the bit that you have, if you're making up a foliar spray, is it neem oil? Yeah, you're making up a foliar spray and you make it to say, I don't know, 300 ppm, and see that, spray it. And if you get any leaves that are twisting or wilting or dying or tip burn, you know half that strength, go back to 150. So then you can work it out for yourself, because you might have a different strength of neem oil that has a higher amount of azadirachtin in them, for instance. And I wouldn't, to be honest, so many people say so much shit these days, it's hard to know. So by doing all these tests yourself, at least then you can eliminate it and you can come back to us with the results. Go, yeah, I use neem oil and I did this. Sweet. We'll just follow what Kincaid, Cassius Clay did. That's my suggestion, mate. But yeah, neem oil's rad. Oh, here you go. Monty's found Viper Soil Testing Code. It's the #1 soil testing. That's what he reckons. Is it legal for foreigners to work in cannabis industry? Is it legal? Well, if you've got a work visa, yeah. But Thailand's a third world country, so I'm sure you can nearly get over there on a tourist visa, and you do all right. I'd be nearly emailing people if I was wanting to go down that route in Thailand. Email some professional companies saying that you're experienced with cannabis, you've been involved with it in however many years, and you're interested in cultivating it for Thailand. Start from there, mate. Send that out to a few different companies and see what you reckon. G'day, Atomic Spoon. How you going? I always thought green algae would add oxygen to the roots. Yes. They actually, aren't they the opposite? Don't they take the oxygen out of the water? How do they do that? I don't know their respiration. I can't honestly answer that without looking into that one. I feel dumb. Oh well, sorry. Would they add it? Because I know that in Australia we get the blue-green algae on the top and it cuts off all oxygen to beneath, so it kills everything. But that might be because does it use it up, or does it just deny access? I think it uses it up. Well, that means that it sucks in oxygen and puts out CO2 like fungus. There's your answer. Oh, jeez. Isn't that funny? Just sitting here talking to myself. Right. Would you raise humidity at all? And would you... What's this for? You have to elaborate a bit, Hooter. Would algae raise humidity? No, because they're not a lichen. A lichen is a moss and an algae, and that sort of attracts and holds in the water. So unless you've got a big... From my algae, I've been doing this for a few years now, and the algae doesn't. All it does is it coats... It ends up going really dark green, and it's nearly makes the pot look black. So I think it's fine. And even when I try not to wash it out, or I'll try and save the green algae after the plant's done, because that's all chlorophyll. That's organic chlorophyll in there, so it can be broken down. Yeah. Brown already is bad. Green algae is good. Brown already is bad. You get brown from the deep waters of the North Atlantic up there, And I know the green stuff they get from off Tasmania here. There's 2 different species of the kelp that they use. Oh, you're talking about algae. Sorry, I don't know about that one. Oh yeah, he did put the algae underneath. Yeah, yeah, they're 2 different varieties, and the brown stuff actually has some good properties in it that are different from the green variety. They're a lot more of a deeper cold water one. And they do something. They've got something different that's housed in them. Kelp sprays are very beneficial. And remember, kelp today, it was given to cows, and it's reduced their methane content by 82%. So it's really good. I do kelp sprays as well, kelp foliar sprays. Remember, foliar sprays are very beneficial for precision agriculture. All right, that's answered that one. Most soil. Do you use Korean natural farming methods? No. I don't. Teas are good. They work really, really well. I use teas. I don't use any protein or anything like that, or any lactobacillus or any milk or any labs in it. I don't try and culture protein or fats. I just try and proliferate microbes, really. Here's something too. When you're making your teas up, did you know that if you want to grow fungus, the fungal spores, if you put it in there at the start, they will germinate and they will grow, and then they'll die straight away. So if you want to culture fungi, you can't do it in the water. Or what I do is I'll put my beneficial fungi in right at the end, before like half an hour, and then I'll take it out then. Tissue culture workshop. Yes, tissue culture is fun as. It takes a little tiny area. Do you know you can make you need a sterile spot. So, it can't be, it's got to be away from all sorts of spores and fungus and everything. Airflow. So a good little way is a tote on the side, and you can transform one of them. So if you get your little engineering glasses out, try and do something with a tote. Put a little fan in the top, UVC light, little candle, extraction. Yeah, cheap way anyway. Rice hulls. There might be an updated paper I can't find. Oh, yeah. Thanks for the stream. Take it easy. Cheers. I hope you're liking it, Lene. It's going a bit long today, to be honest. Dr. Nexus isn't here. These questions are good. I want to understand trichoderma on a deeper level, but can't find any good sources. How can I develop a deeper knowledge? Trichoderma is really good for an inoculant. Why? It targets. There's a reason why it says you add trichoderma to combat this. I'd have to go back into my botanical section for that. There's quite a few different trichoderma species as well that are good for different things. That's the last question. Oh, why did the other soil links put in disappear? I don't know, Monty. Oh, links. I think for links, you've got to be monitorized for putting links. Eat CO2 and respire like plants. If they're green, they eat CO2 and respire O2. No, it's not right. Shamanic dose. How the hell can you flush drainless pot? Salt build up bad. You put water into it, mate. Use reverse osmosis water, because that's got a cation exchange that will grab things because it's got a negative charge. So, or if you're really full on, use bloody floor cleaner. Industrial floor cleaner, put that in. That will strip the living shit out of your plant. Make sure you redose it up with a nice little feed afterwards. 800,000 ppm mix of feed to replace what you just hardcore annihilated out. And the floor cleaner will probably get rid of a bleach, or it'll be a heavy alkaline, so it's got rid of all of your microbes as well. If it was me, I would stick with a reverse osmosis flush by using the cation exchange. Test, put more in, test, put more in, repeat repeat until ppm is under 1,000. Trichoderma, I left it on screen because I was going to try and find you a thing, a bit more stuff on it. Is it trichoderma or trichoderma? Oh, it's trichoderma. Trichoderma is a fungus. I thought it was trichogramma, and that's why it was a bit more technical. But it's a type of fungus, and you want to use that to combat that I couldn't tell you. Sorry. Dave 69. Okay. The only cannabis you consume in Thailand is under 2%. There is no legal recreational use. I don't know about that. There's no legal recreational cannabis. Yeah, I reckon there is. I think it's above 2%. I don't think they've limited the 2% thing. Do algae photosynthesize? Fuck yeah. Yeah, they've got chloroplasts in that do the transformation. And that's why they go green, because of the chlorophyll that they hold inside of the chloroplasts. So that's why we're talking Arthrospora platensis, the stuff that I grow. That's what I try and do. I'm trying to culture that and get it to work for me. The last few days, or it will drown. I think they're talking about adding beneficial fungi to a tea. You add it for the last few hours, last 30 minutes, because if you put it in a petri dish or put your beneficials in water and watch, they'll germinate fast, and then they'll try and grab on and try and proliferate. But if they're drowned, if it's over 100%, or if there's nothing to grab onto, they won't keep sprouting because their germination conditions aren't met. Shamanic dose. Oh, yep. Just that chat. Yeah, they do. I just Googled it. 5th give off sugar and oxygen. Okay, so American ones just checked up. Thank you. And 5th of algae give off sugar and oxygen. What? That sort of says photosynthesis. Oh, I already said that. Oh, that's it. It might be easy to get on the medical though. I'm not sure what that refers to. All right. Great amount of questions today. I hope I've helped some people, and I enjoyed it. Good on yous. And look how long it is. Shit. Thanks very much. Save your question for next week, because it's been well and truly long enough today. It's been the longest one I've ever done. So thanks for your lovely questions. I enjoyed it. I hope you enjoyed it too. And next week is nanotechnology in agriculture. So I'm going to go through some really cool findings about nanotechnology and why precision agriculture is a must, and it's the future. And I'll give you some experiments you can try, some experiments I'm trying, and then I'll elaborate. I'll talk about some of the latest cannabis news like I did this week. There was another cool thing. I'll talk about that next week, just some other cannabis stuff that's happened. So I appreciate everybody coming in here and throwing the questions around and stuff. It's really good. It reiterates my knowledge, and I really enjoy helping people too. So good on you guys. So thank you. Thank you. I'll shout out to everybody in chat. Thank you again. And I'll speak to you all next week. Happy breeding, happy growing, and good health to you all. Bye-bye.